Hi, I'm Laurel Brigham. I'm a third-year PhD student at CU Boulder, and here we are at Niwot Ridge, Colorado, my field site. My work focuses on the effects of biotic microclimates on species establishment in the alpine. You're looking at artificial shrubs, a subset of the plots that I'll be talking about today. The alpine and subalpine are typically thought of as two distinct thermal environments. However, this might be an oversimplification. Heterogeneity in the landscape, such as that due to topography, can lead to pockets of temperatures that might not be typical to the general trend. These pockets, or microclimates, lead there to be an overlap in the temperatures experienced in the alpine and subalpine. Subalpine shrubs, which also create microclimates, are moving uphill into the alpine at a rapid rate. At Niwot, shrubs have moved uphill in elevation 14 meters in 36 years, from 1972 to 2008. The microclimates they form provide wind protection, snow accumulation in the winter, and shading during the summer. This results in a microclimate that offers greater protection from exposure to the elements. A protected microclimate might facilitate the movement of other species that are more accustomed to less harsh conditions. For example, the movement of subalpine species uphill into the alpine could be facilitated by subalpine shrubs that have already made the move. I was able to create the microclimatic conditions produced by shrubs by building artificial shrub structures out of rebar and hardware cloth. These artificial shrubs are designed to trap snow on their leeward side in a way similar to real shrubs. I also attached shade cloth to mimic the effects of shading by the shrub canopy. There are 7 blocks containing a factorial combination of artificial shrubs and neighbor removals. For the neighbor removal, the above-ground biomass was clipped to eliminate above-ground competition, another force other than climate that could prevent subalpine seedling establishment. Additionally, we established 7 pairs of plots in conjunction with real shrubs on their leeward side. Finally, we established plots in locations just downslope of the artificial and real shrub plots. These locations contained populations of the species we were interested in. In the instance that no seeds germinated, these plots would help us to understand whether our results indicated a treatment effect or whether we didn't meet the seed's germination requirements. We seeded over 10,000 seeds across these 52 plots. The subalpine species we seeded were Gentiana parrii, Calamagrostis canadensis, Pulmonium pulcherrimum, and Erigeron glacialis. The final species we planted was Deschampsia cespitosa, an alpine grass planted to serve as a baseline to which we could compare the germination results of the subalpine species. We measured soil moisture and temperature in order to relate the microclimatic conditions to the response of the planted seeds. We also measured natural recruitment, which would help us to understand the baseline levels of seed germination in the plots. Finally, we measured germination and seedling survival of the planted seeds. This data will help us assess the consequences of the uphill movement of subalpine shrubs into the alpine. If the planted subalpine species are able to germinate and survive, this may indicate that shrubs could hasten the uphill movement of those species and perhaps species with similar tolerances. I'd like to thank the undergraduates that helped to plant those 10,000 seeds and collect soil moisture and temperature data. I'd also like to thank my funding sources, including the Indian Peaks Wilderness Alliance.